Okay, so we can start. Welcome everyone to today's edition of Tissue Talks. As you know, you can post your questions during the presentation using the Q&A feature that you see at the bottom right of your screen, and then we will have a discussion at the end of the presentation. It's my real pleasure to introduce Professor Song Lee, who comes to us from UCLA, University of California, Los Angeles. So Song trained in mechanics and engineering science at the Beijing University. At the same university, he obtained a master's degree in biomechanics, and then he moved to the US to one of really prime institutions, University of California, San Diego, to obtain the PhD in bioengineering. And his mentor was Shu Chen, who is a real legend in our field, as I'm sure most of you know. His faculty career started at UC Berkeley, where he spent 15 years as a professor of bioengineering. And then he was recruited to UCLA as a professor, as a department chair for bioengineering, and also as the director of Bioengineering Institute for California. Song is very well known for his research, which is nominally in the field of cell and tissue engineering. And effectively, he really works at the interfaces of multiple disciplines, including, in particular, engineering, biology, and medicine. In his own words, his lab has the overall goal, long-term goal, to unravel mechanisms of molecular and cellular regulation in various contexts. They are conducting research, but they're also very interested in translational studies and development of innovative technologies. His lab is currently working in, I would say, 5 different areas. One is cell engineering in the context of mechanobiology, cardiovascular bioengineering, tissue engineering in situ using stem cells, drugs, and biomimetic materials, immune engineering, and theranostic tissue engineering. Theranostic stays for like a methodology that provides you both diagnostics and therapy in the same session. His work is very well read and very highly cited, and his translational efforts are supported by the UCLA Innovation Fund. So not surprisingly, he has a number of recognitions and awards. I will just mention a few. He gave an honorary Shu Chen lecture, which is very famous. He was a chair of the National Biomedical Engineering Society Conference. He holds the endowed professorship. He was elected to the Academy of Medical and International Academy of Biological and Medical Engineering. He is also the fellow of the Biomedical Engineering Society and the fellow of the American Institute for Medical and Biological Engineering. Song, thank you so much for being with us today, and then you can take over the control of the screen.
Thank you very much, Gordana, for the kind introduction. It's really a pleasure to be here and share with everyone some of our recent research work. Let me share the screen. Okay, so today I'm going to talk about some of our work on the regulation of the cell memory. As Gordana mentioned, the research in our lab is focused on cell engineering and mechanobiology. On the one hand, we are interested in understanding how biophysical factors in the microenvironment could regulate various cell functions, such as stem cell differentiation. And recently, we are interested in understanding the mechanism of cell reprogramming. On the other hand, we are trying to translate what we learn in vitro into potential therapeutic applications for tissue regeneration and disease therapy. And currently in our lab, we are working on neuromuscular regeneration, cardiovascular regeneration, and cancer therapy. So today I want to talk about some of our in vitro work on cell memory regulation, but also there's another example of in vivo applications. So when we talk about cell memory, there are many different types. There are some memory that are short-lived, which is reversible. For example, the cytoskeletal structure, focal adhesions during cell migration, or transcriptional regulation. It could go up and down in response to the microenvironmental factors, or sometimes metabolic state changes. But there are also some cell memory which it could be long-lasting and not reversible. For example, the epigenetic memory that maintains the information that for cell identity, and these will not change even after cell dividing. And immunological memory is another type that is well known, and then in addition, neural memory in the brain. So these are all examples of long-term memories. And today I want to talk about two examples. The first one is on the epigenetic regulation by the viscoelastic matrix. And the second one is on the viscoelastic material regulation of the CAR T cells, in this case, in the context of cancer therapy. So as we know, in the cells, the chromatin is organized into two major different structures. One is the euchromatin, which is the open structure of the chromatin. And in this domain, the genes are subjected to the regulation and could go up and down any time. On the other side, heterochromatin, which is the closed structure in this part of genes, because lack of accessibility for the transcriptional factors, this closed chromatin structure will turn off all of these genes. And this open and closed chromatin structure will determine the genes that are expressed in each cell type and determine the cell fate. So in the past, when we talk about mechanical regulation of genes, especially, for example, fluid shear stress or mechanical loading, and some of these gene regulation are reversible and mostly happen in the euchromatin. So these genes can, for example, with the translocation of transcriptional factor or activation of certain signaling pathways in the genes could change in response to all the factors very quickly. But if you talk about stem cell differentiation or cell reprogramming, whether cell identity needs to be changed, you are involved the conversion between this open and closed structure. So for example, during stem cell differentiation, the stem cell genes that represents the stemness of the cells in the open chromatin will be closed, and the cells will differentiate into specific cell type, and then specific lineage genes in a heterochromatin will be open. And same thing for cell reprogramming. When we want to convert one cell type into another, and we need to achieve the conversion between euchromatin and heterochromatin. So in the past, people have shown that various factors can regulate cell reprogramming. Growth factors, transcriptional factors, small molecules, etc. And the question we asked in the past 15 years or so was whether biophysical factors, such as mechanical factors around the cells, can regulate this process to help the cells to transition from one type to the other. Right? As we've shown here in the Wendington landscape, when the cell differentiates, it could be described as a cell rolling down this slope with hills and valleys. In all of these points, during this differentiation, it's a transient state it's unstable. And when the cell is fully differentiated into a certain lineage, you will reach a stable epigenetic state and also phenotype. So now we are trying to understand whether biophysical factors can help cells overcome this barrier, epigenetic barrier, to be converted from one to the other, or all the way back to the pluripotent state. So here, this circle describes essentially all possibilities when we convert one cell type into another. So one thing that is widely studied is the stem cell differentiation. For example, the pluripotent stem cells can differentiate into multipotent cells, and then fully differentiate into mature cell types. And on the other hand, with Yamanaka and many others' work, we now know fibroblasts or any other differentiated cell types can be converted into pluripotent cells, or it is possible to convert one cell type fully differentiated into another cell type. And this is, we call it direct conversion. So there are many applications for this cell type manipulation or cell reprogramming. And one is you can generate the cells for disease modeling in vitro in drug screening. On the other hand, you could also use this approach to convert one cell type into another in vivo for tissue regeneration. So on the right side is one example. In this case, we collaborate with Kerchen Lab in Colombia. We are trying to do is achieve in vivo reprogramming of the fibroblasts in the ischemic heart and convert these fibroblasts into cardiomyocyte to help regenerate part of the contractile function of the left ventricle in the heart. So in the next few slides, I want to show you an example of how we use this direct cell reprogramming model to study the effect of mechanical cues on the reprogramming process. The reason we use this model is that when you introduce the reprogramming factors, this reprogramming process will happen relatively quickly compared to the differentiation process. Within a few days, the cells will have a dramatic change in the epigenome that will help us to have a better understanding how the mechanical factors could affect this process. So one question is how the mechanical factors will regulate the epigenome or the cell type conversion. There are a couple of possibilities, and one is the mechanochemical transduction. For example, the mechanical factors can regulate signaling pathways, transcriptional factors, and together with the introduced reprogramming factors in regulate the reprogramming process. The other possibility is the direct effect of the mechanical forces. So a few years ago, actually 3 years ago, I gave a talk for this teacher talk series. I described this example. So when you use mechanical perturbation to treat these cells, and you can use various ways. For example, single cell level, you could use micropipette, AFM, or optical tweezers. But we chose to use microfluidic channel in a previous study because this will allow us to process millions of cells in parallel, and then we can observe the effects of the mechanical cues on the cells. So in this case, you can force the cells through the microfluidic channels with different sizes. In this case, you can apply compression forces to a different degree. You can also control whether it's just a one-time stimulation or it could be cyclic. So in this example, we apply the force for milliseconds. So this is almost the same as the ion channel opening, closing, millisecond mechanical loading. So the idea is to see whether the mechanical force can directly affect the epigenome in chromatin organization and then regulate the cell reprogramming process. Indeed, we observed some changes that could be independent of this chemical enzymatic modification of the chromatin. And in this case, we slow down the process. We force this cell into the channel, and we use a FRAT probe developed by Peter Wang's lab at the USC. In this case, it labels the heterochromatin mark histone 3 lysine 9 trimethylation. And the high level is in red, and when the level decreases, it turns green. So you can see here, even though we decrease the pressure and push the cells through the channel, within a few minutes, you can see this heterochromatin mark decrease dramatically. That shows this mechanical effect could be direct. And in one experiment, we show that if you label the laminin associated domain in green and the laminin B1 in red color, you can see the colocalization decrease after cells being squeezed through these microchannels. So that shows that there may be some decoupling between the nuclear lamin structure and the chromatin. So we know that many of these heterochromatin structures are associated with the nuclear lamin structure. And that provides one explanation how the mechanical force can directly impact the chromatin organization. And then we study another example. In this case, it could be a chronic effect of mechanical factors. In this case, it's a passive mechanical property of the matrix. And is widely studied. The stiffness effect of on the chromatin organization was published last year. So I just want to show you two figures to summarize the findings. So it is interesting when you culture the cells on substrate with stiffness, we observe this effect on the cell reprogramming efficiency. In this case, we use the model of converting fibroblasts into neurons by introducing the reprogramming factors. In this model was established by Warnick's lab at Stanford. So when you wait for a couple of weeks and count the number of neurons, we found that the reprogramming efficiency is significantly affected by the stiffness. And this is also biphasic regulation instead of monotonic regulation. And another interesting thing is that no matter what kind of matrix proteins you use to coat the substrate, in this case it's polyacrylamide gel, it doesn't matter. So mechanical factors seem to dominate this reprogramming process. So here you can see that if it's on a stiff surface, you do have some slight increase of cell reprogramming. And then on a 20 kilopascal, you have the maximum level of reprogramming efficiency. So on 1 kilopascal, which was reported before for neural stem cell differentiation, the efficiency is supposed to be higher, especially for neural maturation. It's not necessarily a high efficiency on this soft surface. So that suggests this reprogramming process is different from the stem cell differentiation. And we perform the ATAC sequencing analysis to examine the chromatin accessibility. And here we just show a part of the genes which are the target genes for ASCL1, which is one of the reprogramming factors. And you can see here, red color means more open structure, and then blue color means more closed. So as consistent with what we found for reprogramming efficiency, on 20 kilopascal substrate, you have more opening for these neuronal genes targeted by the reprogramming factors. On 1 kilopascal, it's still better than the hard surface, but compared to 20 kilopascal, it's not as much opening as at this intermediate stiffness. So why so? We perform a series of studies to figured out one mechanism that accounts for this effect. So on the right, we just show a few examples. If you use the tunable hydrogel, when you shine a light to the matrix, you can either increase or decrease the stiffness. If you look at a translocation of the hat, the histone acetyltransferase, and together with other cytoskeleton components, either actin or covalent. And we observe some interesting phenomenon, which is also biphasic. So this is a summary of the finding. If you look at the reprogramming efficiency, we have this biphasic trend at the intermediate stiffness, about 20 kilopascal. We have a highest efficiency. And then, interestingly, for the it's highly correlated with the hat, the histone acetyltransferase activity. And this activity also shows a biphasic trend. And this results in the biphasic histone acetylation in this case, accounts for the enhanced reprogramming efficiency. So this biphasic effect could be due to two factors. One is the actin organization, because on the hard surface, you have more actin stress fibers, but less G-actin monomers. And then G-actin monomer in this case is important together with covalent, will serve as co-transporter for the hat to go into nucleus. So when you have a hard surface, you don't have much monomer of actin available, right? So on a softer surface, you do have a lot of these available. However, another limiting factor is the nuclear transport. On a soft surface, the nucleus is smaller. Also, the nuclear importers, the nuclear transporters, in this case, the importing line is suppressed. That becomes another limiting factor. So at an intermediate level, we could achieve this optimal level of hat translocation to nucleus to induce histone acetylation and enhance cell reprogramming. So this is an example of mechanochemical transduction through the enzymatic regulation. So now I want to talk about the viscoelastic effect. So as we know that in a native tissue matrix, we have water content, we have dynamic bonds. All of these could account for the viscoelastic effect. And viscoelastic material is different from the elastic, as you can see here. When you have a different stretch, loading, unloading curve, and you have you can use loss modulus to describe the elastic part, and the storage modulus to describe the viscoelastic part. And this is a characteristic of all native tissues, as shown in this review in Nature a few years ago. And one thing we use to describe the viscoelasticity is to look into the stress relaxation. In the relaxation time, when the stress is relaxed to about half of this level here, we can use that to as a one quantitative parameter to describe the viscoelasticity. So we use this alginate gel system developed by David Mooney's lab many years ago to study how viscoelastic surface can affect the epigenetic state of the cell and then cell reprogramming. And here, covalent cross-linking is used to control the elasticity and the stiffness, and the ionic cross-linking with calcium ion is used to control the viscoelasticity. And we have a characterization of the stiffness and the viscoelastic property of the substrate. So we first look into the dynamics of cell nucleus. In this case, we label the cell nucleus with GFP lamin A. And you can see on the left is the viscoelastic surface, 2 kilopascal. And on the right is the viscoelastic surface. On this viscoelastic surface, you can see that more wrinkling on the nuclear laminar. In addition, you see more dynamic deformation as well, suggesting there may be more dynamic forces applied to the nucleus when the cell is moving on a viscoelastic substrate. And then we use photobleaching FRAP to measure the chromatin dynamics in the cells. In this case, on the top, we photobleach the DNA DAPI staining. And on the bottom is HQ1 GFP, which was also bleached. And we studied the recovery of this fluorescent signal and use that to quantify the dynamics of chromatin. In both cases, we observed that the viscoelastic surface increase the chromatin dynamics. In this case, the recovery time is much lower than the purely elastic surface. In addition, we stain for chromatin and quantify the chromatin compaction. In this case, it's DAPI staining. And we could see that on the viscoelastic surface, in general, the DNA compaction is decreased. And now we look into the transcriptome in trying to understand what are the gene expression changes induced by the viscoelastic surface. So here you can see in the PC plot, you compare two cases, for example, elastic, 2 kilopascal and 20 kilopascal. And then viscoelastic surface, the same stiffness, 2 and 20 kilopascal, with a slow relaxing, which is hundreds of a second for the relaxing time. It's interesting, you can see here, the elastic effect is shown clearly. These are triplicates, right? 3 samples for the 20 kilopascal and 3 for 2 kilopascal. They are very different gene expression profile. And viscoelastic surface has some effects, and they are in a different direction. It's the PC1 domain. You can see they are all clustered together. And one thing interesting is that they mask the effect of the stiffness. So even for soft surface, right, when you have a viscoelastic effect, in the cell gene expression profile are similar for 2 and 20 kilopascal if they have the same viscoelasticity. And there are many changes of these genes, and this was published as a preprint in bioRxiv last year. So here I just want to show you one thing that is very interesting, which is the nuclear skeletal genes. And here you can see these are triplicates of each condition, 20 kilopascal viscoelastic, and this is 2 kilopascal. So in general, for these nuclear skeletal genes, the viscoelastic surface decrease many of these gene expression. Right, so you can see some of this lamin A, for example, is here. And then for the elastic surface, you do observe a slight decrease when you have on a soft surface. You have slight decrease of these nuclear skeletal genes as well, which is consistent with what have been reported previously. But the most striking thing is that the viscoelastic surface has more dramatic effect than the soft surface. Indeed, this can be verified by gene expression or Western blotting analysis. And we show here as example, lamin AC, indeed, the protein level decrease. In contrast, lamin B1 did not have obvious change. And now the question is, are there any epigenetic changes in the nucleus? In here, we look into a few euchromatin marks, histone 3 acetylation, H3K27 acetylation, and H3K4 trimethylation. In all these cases, we observe the increase of this euchromatin mark. In addition, if you look into the enzymes that regulate this histone modification, for example, for histone acetyltransferase or deacetylase, the deacetylase show a significant decrease, which is consistent with the increase of acetylation. And on the other hand, the histone methyltransferase and demethylase, they change in the opposite direction. That could explain the difference for H3K4 trimethylation. We also performed the ATAC sequencing analysis similar to what we did for the stiffness study. In this case, we found that in the promoter of some neuronal gene, for example, ASCL1 in the MIT1L, you can see some difference induced by the viscoelastic surface. And this is more striking. You can see more increase in opening for MIT1L in this case. And if you look at the multiple genes targeted by ASCL1, which is one of the pioneer reprogramming factor, you can see that some sites closed and some sites more open. And if you examine the reprogramming efficiency, converting the fibroblast into neuron, you can see we have a slow relaxing gel, in this case, more higher reprogramming efficiency. And then if you look at the short-term effects, the gene expression, the neuronal gene expression, endogenous ASCL1 also increased dramatically at day 2. And the mature neuron, if you count the mature neuron as a readout, is consistent with the 1-week result. You get more mature neuron markers expressed in the cells, and there are more reprogrammed cells on the viscoelastic surface. So this is also can be also generalized by different kind of reprogramming model. For example, if you're trying to turn fibroblasts into induced pluripotent stem cells, with the similar result, we found there's some differential regulation of the chromatin accessibility by the viscoelastic surface. And the efficiency of reprogramming, converting fibroblasts into iPS cells on viscoelastic surface also increased dramatically. And this can also be verified by the differentiation capability of these reprogrammed iPS cells. So we have shown that the decrease of a lamin by viscoelastic substrate, and we wonder whether this is related to all of these epigenetic changes in the improvement of cell reprogramming. So we did an experiment to knock down lamin AC, and this knockdown was quite effective. And indeed, if you knock down lamin AC, you can partially mimic the effect of viscoelastic surface. So in summary, for this part, we observed that stiffness can regulate the epigenetic state in the reprogramming process. In intermediate stiffness around 20 kilopascal was optimal. And when you have cells cultured on the viscoelastic surface, there are some additional effect. And it's interesting that the viscoelastic matrix decreased the nuclear lamin AC expression and decrease the chromatin condensation and increase the chromatin dynamics. And this resulted in increase of the accessibility in the chromatin, and then improved the cell reprogramming efficiency. Okay, so now I want to talk about the second story on the immune cell regulation. We know that mechanical factors can regulate immune cell functions. For example, neutrophils, macrophages, and T cell. And today I want to focus on the effect of antigen-presenting cells on the T cell activation. So the reason we look into this is because that it's important for cell manufacturing. We all know that CAR T cell is a promising therapy for cancer. So in this case, it's a 2 weeks process to expand cells outside the cell body. The peripheral blood cells extracted from the patient, and these cells can be activated in a bioreactor. In this case, commonly used method is Dynabeads, which is polystyrene magnetic beads coated with the antibodies, anti-CD3, anti-CD28 to activate T cells. And then you introduce CAR into these cells and further expand them. 2 weeks later, you harvest the cell and inject them back into the patient. However, there are several problems. So this cell manufacturing process is not well optimized, and a lot of space for improvement. One is the donor-to-donor variation. From different donors, the cell expanded have different qualities. And another issue is the persistence of this therapy, which is dependent on the percentage or subpopulation of the T cells. If you have more T memory stem cell, we could improve the persistence of this therapy and improve the durability to prevent cancer recurrence. So one issue in this case is the low percentage of the T memory stem cells in a T cell product. And the other one is one CAR was introduced into these cells during the cell manufacturing process. At the end of 2 weeks, in general, only about half of the cells express CAR, and the other half probably could be negative. And if you inject too many cells without a specific targeting signal, you induce more cytokine storm or side effect without achieving the therapeutic goal. So we are trying to improve this cell manufacturing process. And one thing we did was to manipulate the mechanical property of these beads. We call it artificial antigen presenting cell, in this case, synthetic viscoelastic activating cell. And what we found is that when you just simply change this mechanical property of this antigen presenting cell, you can improve the cell manufacturing process to increase the subpopulation of the T memory stem cells and enhance killing. In addition, we found something unexpected. We can improve the CAR transduction into the cell as well. So we use this microfluidic system to fabricate the alginate beads, and this is the same system we use to manipulate the mechanical property of the microbeads. And you can see here, when you have these Dynabead interaction with T cell, this is SEM images, you do have some immune synapse formation. However, with these viscoelastic beads coated with antibody to activate T cell, you see an extensive immune synapse formation. So this was one interesting observation that we are trying to investigate for the mechanism. So we perform a flow cytometry analysis to determine the subpopulation of these expanded T cells. And this shows the cell manufacturing process. And at day 3, and CAR lentivirus was used to introduce CAR into the cells. And then we analyzed the cell products at a different time point. Here just shows the the data for the time point after 2 weeks. Now, something interesting here. One is that the cell number didn't change much if you compare the Symvac to Dynabeads, but elastic beads without viscoelasticity did not give us a very good expansion efficiency. Another difference is that the viscoelastic beads induce more CD8 positive cells, and this could enhance the T cell, for example, the effector T cells. And on the other hand, we decrease the regulatory T cell subpopulation in this cell product. And another point is that what we found is if you look into the T memory stem cell population, we see a significant enhancement when you use viscoelastic beads to stimulate the T cells for cell manufacturing. So if you examine the percentage of cells that are positive for CAR, and you can see here the Symvac viscoelastic beads induce actually allow a higher transduction rate for CAR into cell close to 90%, while Dynabeads and elastic beads are much lower. So if you collect these T cells after 2 weeks, in this case without sorting the whole cell product, and you can incubate that with different kind of a tumor cell line in vitro and look into the killing effect. In all the cases, if you just look at this overall effect of the cells, the Symvac with CAR has a best effect in killing in vitro. To better understand the subpopulation of this T cell expansion, we performed a single cell sequencing. It here shows various clusters of T cells, CD8 and CD4, etc. I just want to point out that for these two clusters, cluster 4 and 9. Cluster 4 is the T memory stem cell, CD8 positive T memory stem cell. In this case, we observe an increase of this subpopulation in the Symvac treated population compared to Dynabeads. In contrast, for regulatory T cells, CD4 positive, this is cluster 9, there's a decrease in the Symvac treated cell, which is consistent with the results from flow cytometry analysis. And then we tested the therapeutic effect in a couple animal models. In here, I just show the B lymphoma model, the blood tumor. And you can see here, without treatment, most of the mice died. This is a whole body bioluminescence signal to show the tumor in the mice. And if you have a Dynabeads, you do kill these tumor cells, but after 4 weeks, in some cases, the tumor cell will come back. In contrast, for these Symvac expanded CAR T cells, and you can clear the tumor and prevent the recurrence. And indeed, if you examine after a few weeks, in where how many CAR T cells remain in the mice, we examine that in the blood, in different organs, spleen, liver, lung, and in bone marrow. If you compare the case with the Dynabeads expanded cells versus Symvac expanded cells, we always have a high percentage of these CAR T cells left in the mice. So with that, we could conclude that if you have viscoelastic beads, in this case, the mechanical property show very dramatic effect. You could increase the CD8 positive cells and decrease regulatory T cells. In the CMVAC could also enhance the car transduction efficiency, which could improve the kitting capability. In addition, we observe a higher population of T memory stem cells, which could improve the durability of the therapy for CAR T cells. So I want to thank all the lab members in the viscoelastic matrix on epigenetic regulation. That project was led by Yifan Wu, a PhD student in the lab, with the collaborations with a local lab in Johns Hopkins in Amy Road's lab at UCLA. And the viscoelastic beads for CAR T therapy was led by Zhuyang Liu and Raymond Young in the lab in collaboration with Charlie Lee, a talented postdoc in Lili Young's lab at UCLA. I want to thank all of you for your attention. Happy to answer any questions.
Thanks very much, Song. This was really, really interesting. So you can stop sharing now. Perfect. Just to quickly introduce our next speaker, who will be Kent Leach, also from California, but a different UC campus. Kent will speak to us next Wednesday about engineering biomaterials to regulate inflammation and enhance tissue formation. So let's start with questions. The first one comes from Gilad London. Terrific. The implication of your work is that you can infer, to a degree, the viscoelastic state of the cells based on their gene expression. The question is, have you performed a systemic inference on existing data? And do you see differences between secreting versus non-secreting cells, for example, B versus D cells?
Okay, I assume this is for the second part of the talk. Yes. Regarding immune cells. Yeah, we haven't done any study on B cells yet, but we are doing more work on the T cell to investigate the mechanisms.
So maybe, Song, just to broaden the question, could you comment on the differences among different types of cells? For example, are the cells that are sort of mechanoresponsive more suitable for these kinds of interventions? Cells that come from different tissue systems, what is it that you have observed so far in terms of diversity of the cellular responses?
Yeah, that's a good question. We haven't done direct comparison for all of the different cell types, but indeed, you're right. So different cell types will respond differently to the same mechanical cues. For example, epithelial cell and mesenchymal cells are quite different. Suspension cells and the adherent cells will be different too. That could be related to the difference in the cell adhesion, cell matrix interaction, cytoskeleton organization, and also the expression level of various cells components, for example, cytoskeleton, nuclear lamin, etc. And for example, the embryonic stem cells will have no lamin AC expression. Upon differentiation, they start to express those. So in a way, when we culture cells on a viscoelastic surface, it seems that kind of revert the cells partially back to that state at least. So indeed, different cell types will be different. You will not be surprised that for different cell types, people report different kind of responses.
Could you identify some of the underlying features, for example, terminal differentiation versus pluripotency, or some other general properties of the cells that would play a role in these differences? Yeah,
so here we're still talking about the cell type effect, right? So yes. Yeah, for fully negative cells in the pluripotent cells or multipotent cells, the structure may be different. So as I mentioned earlier, one is for example, big difference in the nuclear lamin expression level, but the cytoskeleton level, for example, epithelial cells, and they're different too. If you have cells spread on the different stiffness and or with and without viscoelasticity, the responses could shift based on the mechanical property of the cells themselves, or how much forces the cells can exert on the surface. And if the cells have very strong contraction, it will be different as well. For example, smooth muscle cells, compared to the fibroblasts, they're also different.
Okay, so Aaron Goldstein, very interesting examination of the differences between elastic and viscoelastic materials. So he asks if you have ever considered that the chemistry and mechanics at the surfaces of the viscoelastic materials may be different from their bulk properties.
The surface definitely is different because when we do conjugation, for example, for 2D surface, we perform the conjugation, but maybe if you go down and deeper in the gel, it will be different. So the surface will be different for sure. There may be some gradient of this chemical effect. But when we make the gel, I'm talking about surface modification by conjugation and surface chemistry, etc. But when you make the gel, it should be a uniform across the thickness of for microbeads is the same.
Okay. There is a question from Yofi Weil. It's related to the nuclear deformation on viscoelastic substrates. So he's interested if molecular crowding within the nucleus may explain some of the mechano-responsive pathways. And a follow-up question is, how can one investigate this?
That's a good point, because for the direct mechanical effect, there are several hypotheses. It could be this decoupling of a certain structure. It could be phase separation or molecular crowding. All of these could affect the organization of the crowding. So we haven't done much, but I know some groups working on that, but for different kind of stimulations.
Okay. A question from Karanvir Saini, great work. He is asking if biphasic regulation can also be applied in 3D settings to the tissue matrices, not only in 2D as you have demonstrated.
We haven't looked into this regulation in 3D, because in 3D there may be some additional physical effect. For example, the physical constraint of the surrounding matrix, and also the cell adhesion density will be increased compared to 2D. I think there may be some difference, but there needs some investigation in carefully to see whether there's any difference.
Okay. He just typed like a clarification that his question was about biphasic regulation of the cell programming.
Yeah. So 3D is different. For example, if you have a spheroid or cells embedded in a 3D matrix, the reprogramming efficiency could change it too. So, but we haven't done the study to vary the stiffness in a 3D and look into the efficiency. That may be interesting because it could be related to the in vivo situation.
Okay, a question from Gilad London. Do you know if the number and quality of mitochondria correlates with the viscoelastic state?
Very interesting question. We haven't done extensive study, but we did compare the metabolic reprogramming you know, for the immune cells. There's some difference. Yeah, so the short answer is there's difference. Yeah, it is interesting that mechanical cues can regulate the cell metabolic state as well.
Yeah, yeah, oxidative metabolism. Yeah, yeah.
Yeah.
Okay, so maybe we are like nearing the top of the hour. Maybe we can just take one more question. Okay, so question from Yuhao Zhai, great talk. How does the viscoelasticity or stiffness of synvax correspond to specific tissue or antigen presenting cell types?
Yeah, that's a good question. In the literature, people have reported antigen presenting cell, the stiffness is between 1 to 10 kilopascal. It depends how you measure it. For example, you know, different measurement, indentation, and others will give you slightly different results in how much force you apply. And the relaxation for the antigen presenting cells in mouse and human are slightly different. I think in general is tens of seconds for the antigen presenting cells. So in our case, the synvac in the example I show, it's about 10 kilopascal. So it's close to the cell stiffness. In relaxation time, it's tens of seconds. So in the same magnitude, but the relaxation time is a little bit bigger.
Okay, Song, one more time, thank you so much for a really wonderful lecture. And thanks everyone for all these insightful questions, and hope to see you next week. Thanks, everyone.
Thank you.